Okay, I'll just like to welcome you to this session of NeuroMatch 3. My name is Elaine, and I'll be introducing the speakers today. So just while people are still coming in, I'll remind you you'd be very welcome to share any thoughts or comments you have on Twitter using the hashtag NMC3. So we've got some really interesting talks lined up on 3 very different topics today, ranging from a fly model of Parkinson's disease to the processing of body expressions of emotion, and then oscillations in inhibitory networks. So the first 2 talks in this session are interactive. So that means the talks will be short. The speakers will take about 5 minutes to introduce their research, and there'll be plenty of time for questions. And the third talk will be a traditional 12-minute talk. So you can use the chat to send messages to everyone and share any relevant resources. And please use the Q&A tool to ask questions from our panelists. For the interactive talks, if you'd like to ask your question on screen, you can type on screen beside your question, or you can raise your hand. And also you can vote on which questions that you find most interesting. So please take advantage of that. And do ask lots of questions so we can get a good discussion going. Okay, so I'll start by introducing our first speaker now, is Arun Shankar, who is a PhD research scholar at the SGS Medical College and the King Edward Memorial Hospital in Mumbai. And he works in the lab of Dr. Rakhi Tripathi. Arun is also an amateur astronomer, a science communicator, and a trekker. And today he's going to be talking about an evaluation of anti-Parkinson's activity of herbomineral avracbasma on rotenone-induced Parkinson's disease in Drosophila melanogaster. Thanks very much.
Thank you, Elaine. So I'll just start with my sharing.
Your volume is down again.
Okay. Am I clear now?
Yeah.
A warm welcome to all of you. So my name is Arun Shankar, and I'm a PhD student from KEM Hospital in Mumbai in India. So I am here to present my topic titled Evaluation of Anti-Parkinson's Activity of Herbomineral abracbasma, which is an Ayurvedic drug on rotenone-induced Parkinson's disease in Drosophila melanogaster. So in spite of all the advancement in technology and research activities in Parkinson's, no cure or therapeutic drug has been yet found which can completely stop the disease. So the treatments which are available right now, they only act on the symptoms. So there are no drugs which reverses or slows down the progression of the disease. So we decided to have a look into the traditional Indian medicine system known as Ayurveda. The reason for going for Ayurveda is very commonly used in India, and more than 10 crore people or 10 million people use Ayurveda in India. So basically, Ayurveda majorly consists of either using herbal drugs for treatment or herbomineral drugs for treatments. Now, the herbomineral drugs are a mixture of metals and herbs, and they are claimed to be non-toxic and nanoparticle in size if you look at the Ayurvedic literatures or the references. So these drugs have been used since centuries, but there are very few scientific data claiming their non-toxic nature. So we decided to use a drug called as abracbasma, which is made up of mica, and it is treated with herbs to form a herbomineral called as abracbasma. Now, when any researcher looks at such drugs, the main concern that anyone would have is the toxicity due to metals present when we prepare the drug. So a toxicity study was first carried out on Swiss albino mice. Then a physicochemical analysis was carried out of the drug. I mean, the drug was completely analyzed using modern parameters in collaboration with IIT Bombay, which is one of the topmost research institutes in India. And we concluded that the drug is non-toxic and it is indeed nanoparticle in structure. Now, the organism which we used for our study was Drosophila melanogaster. It is commonly known as fruit fly. The chemical which we used to induce Parkinson's disease was rotenone. Now the rotenone, it acts on the F1 particles of the mitochondria, which leads to the loss of dopaminergic neurons in Drosophila brain, leading to loss of motor ability in flies, and also it leads to reduction in the dopamine level in Drosophila brain. So, and the concentration of the drug was carried out after undergoing an extensive study of viability assay in our lab. The variables which we used to check for Parkinson's disease was climbing assay, from which we'll get to know about the climbing activity or the motor ability of the fly, dopamine estimation of Drosophila brain, protein estimation, and malondialdehyde and glutathione estimation. Now, this is the model which I used. In this, I had 4 separate groups. One was the control group, which had healthy flies. The other was the disease group, in which the Drosophila was fed with rotenone for a period of 7 days, and the concentration used was 250 micromolar. The third group was positive control, wherein the disease was induced, and along with the inducing agent, L-DOPA was given, which is a golden standard for Parkinson's disease. And in my final group of study group, I used the herbomineral at a concentration of 0.25% of the total medium. Now, and on the 8th day, we carried out certain parameters such as the climbing assay and estimation of dopamine and protein levels. So it was clearly observed that there was a marked or significant improvement in the climbing assay, in the motor ability of the Drosophila flies in the group where the abragbasma or the herbomineral was given. And there was a significant improvement in neurotransmitter level also. The dopamine levels were estimated using HPLC, and there was a significant difference when we compared it with the disease control group. And when we look at the inflammatory markers, there was an increased or an improvement in the malondialdehyde and the glutathione levels in the groups which were fed with the abragbasma group or the herbomineral group, and also a protein level was observed to be improved in the study drug group. Now, certain parameters are being standardized in our lab to further study the Parkinson's model, and these are flow cytometric assays to check for the measurement of reactive oxygen species generation. We are also planning an experiment and standardizing photoluminescence assay, wherein we will be looking at the ATP activity of the mitochondrial cells. And we are also undergoing immunohistochemistry of dopaminergic neurons of Drosophila brain. Now, the following conclusions were made after carrying out the assays. It clearly showed that the abragbasma or the herbomineral group helped the Drosophila flies to restore its motor ability. It also helped to increase the level of dopamine levels in the study drug group when compared with the rotenone or the disease control group. There was also an improvement in the malondialdehyde and glutathione, suggesting that there is an anti-inflammatory property of the drug. And this also proved that the abragbasma or the herbomineral drug can be used as a potential drug for treatment of Parkinson's disease, but further studies are required to validate the claim. Thank you for for listening to my talk. And also, I would like to add 2 or 3 more points regarding this thing. It is very important to look into the new systems when we are unable to find out a therapeutic drug for any particular disease. And when we especially look at the traditional medicines of India or any South Asian drugs, it is very important to check for its toxicity, because metals are very commonly used in those drugs. And scientifically proving that it is non-toxic and it has a very good efficacy will be a big boon in countries like India, because these drugs are very commonly used, and there are millions of people who depend on such drugs. Thank you.
Thank you very much, Arun, for a wonderful talk. So I'll wait for any questions to come in. So remember, please, if you'd like to ask your question on screen, then type on screen beside your question, or I'd be happy to read it for you. So maybe while we wait for questions, Arun, could you elaborate a bit on the neural mechanisms of the herbomineral drug?
Actually, that's an area which I really want to focus on. The problem is that these herbomineral drugs or the Ayurvedic drugs, they have been used since centuries, since thousands of years. but it's purely based on the Ayurvedic literatures. There are no scientific data available on the same. So right now we are trying various methods on animal models first, and we're also planning to work on stem cell cultures to learn more about the activity, how it acts on the nervous system and all these things. So it's an ongoing study.
Cool. Okay. And could you, would you like to maybe discuss the possible benefits of the drug over L-DOPA, which is the standard treatment?
Yeah, if the drug is really beneficial and it is non-toxic and it has a good efficacy as claimed by the literatures, the good thing will be that its main thing is it is a nanoparticle. So it can easily cross the blood-brain barrier and reach the brain more quicker and more efficiently as compared to L-DOPA. And the second thing is a very minute amount of drug is required. Like if you compare, when you give L-DOPA, majority of the drug doesn't reach the brain. But when you give herbomineral, it has been proved in certain studies that it almost 90% of the drug which we have, it reaches the brain. So it has got a higher level of crossing the blood-brain barrier, which leads to lesser amount being introduced into the person, and it is cost beneficial. And it can also be, what do you say, it's, the main reason is cost benefits, because India as such is not quite a rich, it's a middle class country. So there are lots of economic problems over here. So these drugs, they're very economic, and if it is really beneficial, it will really help the lower middle class family and the poor family in the long run.
And I was wondering, are there any, sorry, I don't know if I pronounce it right, but any Ayurveda drugs that have been tested with human participants in any other neurodegenerative diseases?
There have been studies on cancer using Ayurvedic drugs, but on neurodegenerative diseases, I don't think there have been much drugs tested. They mostly test herbal drugs. There have been studies of herbal drugs on animal models, on rats and mice. And since last 2 to 3 years, studies have started coming up on stem cells and tissue cultures. But it's like it will take more 10 to 15 years to get a proper idea of all these studies. Plenty of studies haven't been published yet. That's the major problem.
That's great. Okay. Well, if there's no questions from the audience, then we might move on to the next speaker. Thank you very much, Arun, again. It was an excellent talk. Okay. So our next speaker is Dr. Giuseppe Marazzo. Giuseppe has a PhD in cognitive neuroscience and works on emotion.
Actually, I'm a PhD student, not yet.
Oh, I'm sorry. Giuseppe is a PhD student in cognitive neuroscience and works on emotion and body perception. He is working in the lab of Professor Beatrice de Gelder at Maastricht University. And today, Giuseppe is going to talk about the dynamics of body category and emotion processing in high-level visual prefrontal and parietal cortex.
So can you see my screen?
Yeah.
Okay, thank you, Elaine, for the introduction. So I'm Giuseppe Marazzo, PhD student at Maastricht University. I work with Professor Beatrice de Gelder in the Brain and Emotion Lab. And today I'm going to talk about the task dynamics of body category and emotional representation in high-level visual prefrontal and parietal cortex. So different studies have provided detailed understanding of how a familiar object like the human body is processed. And we also know that the brain encodes stimulus information in a high-dimensional representational space. This encoding process appears to be dynamic, relatively task-sensitive, and it also may be at service of different and complex behavioral goals. What is actually less clear nowadays is to what extent specific body attributes like the emotion, for instance, might influence the activity of these body areas, and also whether the task performed may impact on the representation of this object category. So in order to clarify this situation, in the present study, we measured the representational dynamics of explicit and implicit body expression perception, and we investigated whether the type of task influences the representation of bodies and body expression inside and outside the body selective category areas. We used fMRI data of 20 participants scanned while they viewed still images of angry and happy body postures of Black, African, and white Caucasian ethnicity. Our experiment comprised 2 categorization tasks over 4 separate runs. Each run consisted of a presentation of, and let me point it out just for clearness. So our experiment comprised 2 categorization tasks over 4 separate runs. Each run consisted of a presentation of an emotion block, as you can see here, and a shape block. During the emotion block, the participants judged whether the emotion expressed was anger or happiness, here at the bottom. And during the shape block, participants judged whether the shape which was superimposed on the body was a circle or a square. For what concerns the analysis, to analyze the brain responses, we used the Univariate GLM, searchlight multivoxel pattern analysis, and representational similarity analysis. On the left side, we have the results of the Univariate GLM when contrasting the activity within the 2 tasks. The color map here indicates the region where higher in red or lower in blue activation was found basically for the emotion recognition task versus the shape recognition task. On the right side, instead, we have the above chance classification accuracies produced by the classifier when trained to discriminate between the 2 tasks. The color map here indicates the t-value of the group pair sample t-test performed against chance level. It is worth noting that ventrolateral prefrontal cortex, IPL, and EBA are among the areas where there is max the difference between the 2 tasks, and also ventrolateral prefrontal cortex and IPL appear to be more active in the explicit task and in the implicit task, respectively. These ROIs are instead the results of the classification accuracies tested against 0 produced by the classifier threshold with a Q of FTR 0.01. On the left side of the panels, we show the representational dissimilarity matrices computed with 1 minus Pearson's correlation distance between the different conditions, which here are labeled in red for the emotion explicit task and in blue for the shape implicit task. On the right side of the panels, we find the group averaged ROI beta values plus the standard error. It is interesting to notice that the representational dissimilarity matrices for ventrolateral prefrontal cortex and EBA present a pattern of similarities within the explicit condition. From this, we see that the difference between explicit and implicit body expression processing can be decoded in these 2 areas, although no emotion specificity there is. On the same page, we see that there is a higher activation for the explicit task in the same 2 areas, while on the opposite, we have a higher activation for the implicit conditions or the implicit task for IPL. So to wrap up, in this present study, we measured the representational dynamics of explicit and implicit body expression, and we identified the brain areas that are critical for the distinction between these 2 tasks. Our results indicate that the task affects body category representation, and in the specific EBA may be active in response to explicit body attribute recognition. Ventrolateral prefrontal cortex may also itself play a role in this categorization because it also codes for body category when the task demands it. And as take-home message, we can say that the combination of higher activation in EBA and ventrolateral prefrontal cortex and respectively lower activation in IPL suggests a connection between these 3 areas, with the ventrolateral prefrontal cortex possibly influencing EBA positively and IPL negatively. Moreover, we can say that the task performed changes the object category representation. And last but not least, depending on whether the emotion recognition is explicit or implicit, different brain areas are involved in the process. So thank you very much. If you want to know more, have a look at the paper.
Thanks very much, Giuseppe, for that's a really interesting talk, a really nice demonstration of how higher level neural representations are context dependent. I was wondering how much of this would you think is specific to emotion processing, or would you find, do you think, similar findings if you had some other body expression discrimination task?
For bodies, it has been shown by Wager and Bugados and Bracci that there is task sensitivity in frontoparietal areas. The most interesting thing is that this task effect is usually confined in frontoparietal areas. So there is no evidence of a task effect in the literature or in a high-level visual cortex, or in more specifically ventrotemporal and ventrotemporal cortex. Let's say that EBA, so specific body areas, didn't show a task effect, let's say, so far. So there is for sure a task effect which is not related directly to emotion, but only the body attributes, let's say, and has been shown by previous studies. Here I also put the references about it.
And I was wondering about whether there was a difference in difficulty across the two tasks, or whether that was
there is I didn't report the behavioral analysis because yeah, I mean 5 minutes it's not a lot, but yeah, in general there is a significant difference, but I don't think it's a substantial difference. For instance, for the accuracy values between the two tasks, we have 96% accuracies for the implicit task and 92%, 90%. I don't know exactly the digits, but 90% for the explicit task. So this difference appears to be statistically significant, but at the end of the day, is not, let's say, substantial. So the tasks were designed to be very similar, in order, I mean, really to rule out possible I mean, differences in the task difficulties.
Did you look at all at whether the difficulty might change over time? Was there repeated exposure to the same stimuli?
I mean, I didn't get it, sorry.
I don't know. I was just thinking about whether if you were repeatedly exposing them to the same images, would people kind of be
sort of adaptation? You mean sort of adaptation? Yeah, I mean this we didn't actually try it because yeah, of course it's always the time that you have for performing the experiment is limited, and yeah. So we didn't actually check for adaptation. We do have a certain amount of repetition of each unique stimuli, of course. I think it's 5 repetition of each unique stimuli, and this didn't rule out, let's say, didn't influence too much, let's say, the behavioral results.
Okay. Well, we had a third talk lined up. If nobody has any more questions for Giuseppe, I'm not sure if our third speaker is here.
Yes, he is. He joined.
Okay. All right. Well. Okay, I'll introduce our third speaker today then.
Thank you very much.
Thank you very much, Giuseppe, sorry. Okay, our final speaker today is Nariman Ruhi. He's a PhD student of computational neuroscience at the Institute for Advanced Studies in Basic Sciences in Zanjan, Iran. And he works in the laboratory of Dr. Alireza Valizadeh. Sorry. And today he's going to talk about the effective transmission delay on the oscillatory behavior of network composed of excitable inhibitory neurons. Are you there, Nariman? Are you there?
Nariman, if you can hear us, I don't think your microphone is connected.
We'll just wait a minute and see. Okay, maybe we'll give him a couple more minutes. Sorry about this. So in the meantime, if anyone has any questions that they've thought of for either of our panelists, please feel free to put them in the Q&A while we wait to see if Nariman can join us. or please leave any comments in the chat.
Actually, I have a question for Arun, if you're willing to answer. One of your discussion points was on looking at new models and systems for studying Parkinson's. What exactly would a new model, like what properties or characteristics should it have? Or what would you prefer it have?
Yeah, thank you very much for the question. So it's like when I look for any new model, well, to be honest, we have a very. Am I audible?
Yeah, you are.
Yeah. Okay. Well, so to be honest, in India, when we do any research, it's really difficult to get the fundings. So when we look for any model, we look for certain models which are easy to culture or maintain in our lab, and even we also look at the parameters which we can do. It's like when I do Drosophila, we mostly go for immunohistochemistry or for basic, behavior assays and also flow cytometry, of which we have got facilities. So I first look at the facilities which we have got, and then we go and select a model. So stem cells isn't like we just established a stem cell culture lab in our hospital. So that made me look into stem cell cultures. So we just started procuring the chemicals and the cell lines. So we just hope we start getting results with it, and we hope to standardize it as soon as possible. Like I work in a place where we have got very less funding. So there are plenty of limitations which we face over here.
Thank you, Arun.
Thank you.
So Nariman, have you got a voice? We can't hear you still. Hello.
Can you hear me? Hello? Hello,
Yes,
Elaine?
hi.
Can you hear me?
Yes.
I do apologize because of my poor network connection.
That's no problem. You can go ahead. We're a bit short on time now for your talk, so please go ahead.
Okay. Let me share my screen and okay. Well, can I start? Do you hear me?
Yeah, I think we can just see your Zoom screen.
Can you hear me?
Yes.
Okay. Okay, that's it. Again, hello everybody and welcome to my presentation. I'm Naiman and in this work, we investigate the collective behavior of emphasis on the function delay and the features of collective behavior of the network. So first, let's state some few points as an introduction. We know from many research that inhibitory instead few points as a action. We know from many feature neurons can generate collective neural stimulation, either by reciprocal interaction by with excitatory neurons or by itself and by its intrinsic mechanism and noisy external drives leads to activation of some of the neurons, and this noise try to generate some burst in activation of networks. And in between neurons try to suppress the networks, and synthesis between these two dynamics and features of inhibitory networks. And I will show you that the transmission delay between the neurons has an important role in the collective behavior, and also I will show you that the networks has a kind of dynamical memory or a temporal correlation in its activity. A few points, all neurons investigated are excitable. We use leaky integrated and fire model. Probability equals to 50%, and all neurons receive a Gaussian noise with variance of sigma, that we name it noise strength. I think this figure, and I want to tell you about our research, and so please pay attention on the top of this figure, and you can see the collective behavior of the network, and clearly you can see an oscillatory behavior of the network in spite of irregular and sparse firing of single neurons, as you can see here in the middle of figure. If I try to explain these mechanisms, I can say that in a time interval, for example, the time interval that here I show you with green color, some percent of neurons find a chance of firing so stochastically, and we name this time interval burst period. After this burst period, and recurrent of inhibition suppresses the networks, and what we see is an silence period of networks activity. Succession of these two time intervals leads to individual neurons fires so sparse and so irregular. And when we have a look on at the population activity of the networks, we can see that amplitude of activity varies in time, and but it seems it is not so stochastic, and I believe there is a temporal correlation, say that the network has a kind of memory of past, and there is an interaction between bursts in the time. So based on the previous slide, as I said, our viewpoint is to split the network activity into the two intermittent time interval, as I said, the burst period and silence period. As you can see, burst period is over average activity time interval and vice versa silence period. And in a burst period, as I stated, some neurons stochastically fire and uh, suppresses the network for a silence period, and it is obvious that some of these two time intervals makes the period of network oscillation, and it varies in time, but not stochastically, and it means the network has the kind of temporal correlation. For first step of our investigation, let's expand the parameter space for various noise strengths and coupling strengths between the nodes, and you can see various kind of dynamical regime in this parameter space in this figure. Variance to mean ratio of network activity. We use this measure as the synchrony index of network activity. It can be obvious that moderate values of noise strengths and coupling strengths can be ideal for us, because in this area, for example, area 2 uh, of networks, emphasize that the network needs enough noise to generate the burst and to be synchronized and to be oscillatory, but excessive noise can destroy the, I mean, so this is first result of here is one expanded parameter space, as I stated before. And this simulation is done for various values. Transmission delays from 2 to 8 milliseconds. Again, color represents the synchronicity between the synchronicity of network activity, and as you can see, increasing the delay enhances the synchronicity of the network activity. It can be obvious because more leads to longer burst period, and it leads to hormones contribute in each burst averagely, and it means the synchronicity of the networks gets larger and get stronger as the result shows.
Man, I'm so sorry, but the connection is just really poor. I'm not sure there's much point in you finishing the presentation because we can't hear you very well at all. So would you like to
Another
wrap up?
important features of the networks is sparseness based on our definition. Oh, I'm sorry. Let me.
I'm
check
so sorry.
something. I'm so sorry because of this technical problem. Let me. Uh, mm,
Because we're running out of time now for this session. So I don't know if you just want to give a 1 or 2 sentence wrap up and we finish. I'm
that's
really
my
sorry. It was
problem,
fascinating
and
work.
I cannot do anything.
Okay, well, I guess maybe we'll finish up the session then. Thanks everybody so much. Thanks to the speakers
Too
for their really
silent.
good talks, and thanks, Narman. I'm really sorry about that disappointing technical issues.
Okay. Can we continue?
Okay. Hi everyone. Rosan, can you back to this session and start for introduction? Can you hear me, Rosa? Rosa, can you back to this session and extract from this slide? Can you hear me, Rosa? You're muted.
Ehsan, I think you are muted.
I'm
I can hear
I'm
him.
not.
Yeah, I
Can
can
you
hear
hear me?
you now.
Okay,
Yes.
let's start and I introduce and start next. Hi everyone, welcome to this session. I'm Ehsan Rezayat, postdoc at the School of Cognitive Science in Iran, and I'm is a MC of this session. And Forzan Farahani is a PhD student at City University of New York, is back end of this session. If any technical problem happen, you can send it to Forzan and she will deal with that. In this session, we have 3 talks. One of them is canceled, was by Nora Bradford, and he sent email and asked me to cancel. The next one is using functional connectivity to predict working memory performance in human connection project, presented by Corey Richard, PhD student in the Heller Lab at the University of Illinois. And I think that we will have a great talk by Corey. Let's Corey to share his screen and start. Thank you.
All right, hello everybody. My name is Corey. I'm here to present a project that was undertaken by me and some group members of mine actually we participated in the NeuroMatch Academy earlier this summer, which was a great experience learning, building some skills in computational neuroscience. So all 4 of us, Ladan, Davide, Nora, and myself are graduate students at various universities in the United States, Canada, and Europe. So it was really cool getting to know and work with everybody. As part of that, we presented a project, or we worked on a project. So what I'm going to do is talk a little bit about that, our experiences from what we learned, and some implications for building models of predicting cognitive performance. So we'll get right into it. So a central goal of neuroscience is to make predictions or understand how patterns of neural activity relate to cognition and behavior. So one common way to utilize this is using fMRI, task-based fMRI, as well as resting state fMRI. A core component of many aspects of cognition is working memory, which is related to a lot of basic as well as clinical outcomes of central interest for my lab, as well as many others. Supervised machine learning is becoming more sort of like a growing method for predicting different outcomes based on aspects of neuroimaging. So things like age prediction, vision, different clinical and psychopathological traits as well. Machine learning has been used to predict working memory performance in EEG in terms of other neural measures, but we sought to use machine learning to try and predict working memory performance using data from the Human Connectome project. So like I mentioned before, all 4 of us met and worked in the NeuroMatch Academy, and as part of our participation, we were provided a subset of the HCP data set with the Glasser parcellation. So we implemented a general linear model approach just to sort of see what would work best using ridge, lasso, and elastic net regression. So ridge regression works to reduce overfitting by shrinking the magnitude of model coefficients, whereas lasso will compress some coefficients to zero. Elastic net utilizes some combination of both of these procedures. So procedurally what we did is we built our models using Python and scikit-learn. We decomposed the Glasser parcellations, the connections to be predictors for a model. We used grid search DB to search for the most effective alpha values for our regularized regression models. And then we fit our ridge, lasso, and elastic net models with a 10-fold cross-validation. Specifically, what we were attempting to predict was predicting working memory accuracy from both the task and rest activity that were provided and seeing which was better. And what we found was maybe ridge regression worked a little bit better in terms of the R-squared value. While the root mean squared error seems promising, we may have had a little bit of a problem with overfitting due to the large number of predictors and not necessarily having a huge number of samples. So in terms of what we learned from this project, task activity was found to be a better predictor of working memory task performance, but only for ridge regression. Otherwise, the models seem to mostly perform the same. So a possible implication for this is that task and rest activity have been found to be pretty similar overall. So that might have some implications for building machine learning models of using resting state or task-based fMRI. Maybe in some cases they could be equivalent. Sort of, maybe it's hard to know necessarily from what we did. And I think another interesting point to be made is the brain linear? Is it, this is sort of like a hot topic of debate, but does it make sense to build linear models? Maybe for some cases it does, maybe for other cases it doesn't. In some other analyses that we did, we found that frontal parietal networks are found to be more predictive of working memory performance, which bolsters a lot of the literature. We use, and maybe it's some things that we could expand upon in the future. So we use functional connectivity as predictors. So that really compresses a lot of the temporal dynamics. Maybe that's where we lost out on some of our predictive capacity, as well as smoothing over the block types of the working memory task in the Human Connectome project actually has a lot of subcomponents that we didn't necessarily investigate. And then, and also establishing accuracy. So we are basically predicting like the continuous score of accuracy. Is it necessarily the most useful thing to predict? Maybe it's more useful to try and predict like how a person will perform on an individual trial or something like that. And then lastly, probably our biggest methodological thing that we had to tackle next time is our problem with dimensionality. So we had way more features than subjects, and that probably led to some problems in building our model. All right. Those are my references. Otherwise, thanks for listening. I guess we have some time for questions, right? For the interactive talks.
Thank you for your talk. And let's send the question in the queue, question button at the Zoom. We will ask them from the lecturer. And also, there's one question that I have, that you found that the performance by frontal parietal network is predicted better. How about the other network or connection? Is there any other network that predict the performance of subject in the task-based data?
I don't think so. I actually didn't. I'm probably not the best person to answer that question from our group. Some of the other group. I'd like to say that our group all contributed to this equally. So some of us were more involved in some processes than others. So I think, and from my understanding, was that was the network that was found to be the most predictive. But I will let, if there are other group members here, they can maybe speak to that.
Okay, there's another question by Hadi Mutamedi. What would be the feature matrix for this HCP dataset?
And feature metrics?
Yeah, feature matrix for this HCP dataset.
Could you maybe elaborate on that?
Can you ask from Hadi if he wanted to ask the question himself?
Oh, what do we train on?
Rosan, can you bring him?
Yeah, so we were training on the parcellations of the. Oh, I guess they're coming in. So if they want to ask their question,
Can you unmute your microphone, Hadi?
Hello, can you hear me?
Yeah, yeah.
Hello, everyone.
Hi.
Great talk, thanks. To elaborate on the question, my question was that, is this, so you did last regression and ridge regression, I think. So my question is, what exactly is the feature matrix? And so our output is, we want to predict the working memory. So we know the labels, that would be our outputs, right? But what are the inputs, the X which is put together in features, which we will regress regressors to come up with the predictions and train our beta values? I don't know if I could be clear about that.
Yeah, sorry, it was clear. I think I just misheard last time. So that was the, we parcellate, uses the Glasser parcellation. That was what was provided for us in the data. And then we evaluated the functional connectivity matrix. And then we decomposed the matrix into the connections, each of the lower diagonal of the matrix. And then that was the features that we used to train on. Does that clarify?
Yeah, right. I get it. But how do we encode the time into this matrix? Do we account for the time variation in the fMRI datasets? Because it's time series, right?
Yeah, so this is where, this may not be the best approach, because what you in effect have to do is compress down a long time. So you're basically taking the average of that connection over the entire time series.
Oh, I see.
Yeah, so it's in essence, the time variation, and this is, I guess I covered this a little bit fast, is sort of lost using that. So using some way to evaluate dynamic functional connectivity. So how the things fluctuate, some variance would maybe probably be a lot better. But hopefully that clarifies. It's sort of agnostic to time.
Yeah, thanks. It's clear now. Thank you.
If there are any other questions that we can ask for you, we have 4-minute time.
I think I will say that Ladon, one of our teammates, hi Ladon, she also worked on this project. She's saying that we tried other networks and the frontal parietal network was the best one. So thanks for jumping in there.
Peter, no. Okay. We can. Is there any other question? Okay, let's take time and go to the next one. As I see, it doesn't come any other question that be. The next lecture is the title is Understanding Transformation Between Decision Potent and Decision Null Representation in Visual Working Memory using DMX PCA, presented by Guan Wang, PhD student in the Dr. Postel lab at University of Wisconsin. And I think that we will learn too much here. Let's start back one, and just please share your screen and start. We have it.
Can you hear me
Yes, yes. You're
and see my screen?
Yes, just put the full. Yeah, it's great.
Okay, cool. Alrighty. So good morning, everyone. In our daily life, we constantly need to juggle multiple pieces of information held in mind. For example, at a cocktail party, you're introduced to Bob and Jim, and you hold these names in your working memory. When you go up to talk to Bob, his name is prioritized in memory. Because you want to talk to Jim later, you still hold on to the name Jim, but at this time, it is unprioritized in memory as you talk to Bob. And later, when you go talk to Jim, the opposite happens. To study the neural bases of attentional prioritization, we use an age-old working memory task called two-back. This task requires subjects to constantly compare the current stimulus with the one that was presented 2 times ago. Let's focus on item N here for an example. Say this is when you first learned the name Jim, and then you wanted to talk to Bob next. So during this time period, the name Jim is an unprioritized memory item, or UMI. And after that, when you want to talk to Jim next, UMI. And you want to talk to Jim next, his name becomes a prioritized memory item, or PMI. To study the neural representation of the stimulus as a function of behavioral priority, in previous work in our lab, we recorded EEG during this task and showed that UMI and PMI are represented in the same code or subspace, but in different and opposite formats. To understand how the representation is transformed between UMI and PMI, we trained a recurrent neural network with LSTM architecture to do this task. Briefly, stimulus information in the form of a one-hot vector goes into these 6 stimulus selective input units. Information is then propagated into this LSTM layer, which in turn projects to the output unit and yields the decision, match versus non-match. To look at the internal representation of the network, we did a PCA on the activity patterns in the LSTM layer and projected them onto the first 2 PC dimensions and mapped them back onto this two-back time course, which we call a trial. The network is run on many trials. Here, each dot is a stimulus representation within a single trial at this time point, and the color always indicates N, or Jim's identity. And we track N's representational structure of all the trials over time. After stimulus N is presented, its representation falls onto these roughly parallel color planes, and the structure rotates roughly clockwise as N transitions from UMI to PMI to finally compare with N+2 to make a decision. In this talk, we mainly focus on these 2 delay periods. To characterize the rotational transformation from UMI to PMI, we used a dimensionality reduction method called demixed PCA. Unlike PCA, DPCA ignores trial-to-trial fluctuations and can tease out sources of variability only dependent upon the stimulus condition, which we care about. It allows us to find UMI and PMI specific subspaces indicated by these black arrows. Some results. Here are the DPCA projections from an example neural network. The axes for each figure are the top 2 DPC components, and the first row is the UMI subspace, and the second row is the PMI subspace. The columns are stimulus response averages based on UMI versus PMI. So in each subspace, notice that the representational structure shown by the order of the colored dots from left to right is very similar between UMI and PMI. Actually, if you rotate the UMI representation clockwise, it will match the PMI data and vice versa. The fact that we get roughly the same representation in each column suggests that UMI and PMI are coded in similar dimensions. Here are EEG data for an example subject. Again, stimulus structures are similar column-wise, meaning UMI and PMI are coded in similar subspaces, consistent with results from our previous work. And row-wise, if I rotate the UMI figures on the left, they can be aligned reasonably well with the PMI data on the right. Just to orient you, for example, in the first row, if we anchor this figure with red, blue, and cyan, we can see red, blue, cyan on the right figure, and the yellow and purple are in the middle, and the green is aligned less well. And on the bottom row, if we align the figure with green, cyan, blue, green, cyan, blue, and again, the yellow and purple are in the middle, and the red is aligned less well. We can see that the alignment of the EEG isn't as good as with the RNN. We think it is because EEG data is very noisy and high dimensional, and the rotation may happen in other dimensions as well. It's also possible that representations of orientation stimuli in the brain are not nonlinear, so they can't be well captured by linear compression of the data. It is still a work in progress to get a bit clearer picture on this, and overall DPCA is tailored to extract signals of interest and should allow us to qualitatively and in the future quantitatively characterize and compare the RNN and EEG representations. I'd like to thank my lab and funding and also my awesome collaborator, Jorge. And here are some next steps and challenges. And I'm happy to take any questions, comments, and suggestions and clarify anything that was unclear. Thank you.
Thank you, Kwan, for your talk. And is there any question by anyone? Can you send your question in the question button and we will ask. No question?
It's probably too early for people.
Okay. There's some question. Jack Stewart asked, I think that is normally the case that the UMI item are harder to decode in PFC, for example, than PMI item. Does the LSTM network display this as well?
Not really. So in the LSTM network, the decodability of UMI and PMR are similar. Yeah, again, it is not the brain, and it probably doesn't have the brain-like representational characteristics. Yeah.
Hadi Motamedi has a question. Hadi, you can unmute your microphone and ask a question yourself. You is panelist.
Okay. Great. Hello, thanks for your great talk. It was really awesome. My question is that from EEG data, it's very cool that what you're saying is we can know which working memory it's capturing, right? It's visual task, if I'm understanding it correctly.
Yes. Mm-hmm.
Yeah. So in this visual task, you can decode from the EEG which working memory or which image actually the participant is seeing and how long, for example, it's keeping it into this memory, something like that.
Right. So you can imagine in the two-back task after the presentation of each item, say the item is item N, okay? So after presentation of N is unprioritized, whereas N-1 is prioritized, right? So you basically, it's the same neural data, but you can decode it using or reconstruct it using the PMI label, which is N-1, or the UMI label, which is N. So that basically can dissociate which working memory it is.
Okay.
Yeah. Okay, thank you.
Thank you, Adi. And another question by Dr. Paulson. Farzan, can you bring him on ask his question?
Oh, yeah, okay. So Brad asks, your RNN only has one hidden layer. How might you describe? Sorry?
Here is Brad is on.
Oh, okay.
Yeah, I was just following up on the question from Jake Stroud, right? I think that's what he's asking. And so the next step, I guess, would be, how would you represent different brain areas? Presumably, you'd need different layers in your RNN, right?
Right. So I would say, so right now it's just LSTM, and it only has one layer, and it doesn't have the distinction of PFC or parietal lobe or occipital lobe. But we can imagine we can possibly extend the network to a more biologically like a realistic network. For example, the well-established PBWM, the prefrontal basal ganglia striatum network. So we can model some features or cell types and of different brain areas and connect them with connections that are constrained by empirical data. And that way we can better tease apart what kind of representation or where the representation lives.
Okay.
Yeah, so one comment, just to continue the discussion on this, one comment I'd make is that right now like Shwan was just saying, the LSTM is just one homogeneous network, and from the perspective of the task that it's trained on, both the PMI and the UMI are equally important. So clearly it's going to be representing both of them equally faithfully. But if we start to add some more structure into that LSTM, where maybe some areas are playing more of a role in doing PMI or less of a role in doing UMI, et cetera, then maybe some of these differences might arise that Jake pointed to. But yeah, I think it's an interesting question.
Okay, and another point that Sanchit Gupta is asked, I might have missed it as I joined halfway. I understand, but she said, can you unmute microphone and ask your question?
Yeah. So I want to ask, like, how exactly do you come up with that DPCA subspace? As I missed your first part, so if you can briefly tell, that would be great. Thanks.
Right, sure. I can show you the DPCA algorithm. Let me just share my screen real quickly. So can you see it now?
Yes, obviously.
So this is the essential DPCA algorithm. So what it does, let me parse it out for you. So here the X bar is the grand mean of the data, overall trials and time points and stimulus conditions. And the XS is the mean response for the stimulus S. So this term captures how the stimulus means fluctuates around the grand mean, or the stimulus specific variance. And here we have the response on time point or trial I for the stimulus S. And this basically shows how each trial or time point fluctuates at the grand mean. And what the DPCA does is that it has a decoder vector and an encoder vector. And the goal of DPCA is basically reduce this reconstruction error, which is the expectation over all stimuli and time points. And this way you can basically capture the variance that's only specific to a certain task parameter. In our case, it's the stimulus. So basically we use this W vector. Well, you can imagine like PCA, you can order the different components by the variance explained. So we can choose the top two DPC components to construct the two-dimensional subspace.
Okay, thank you. And what is the size of X? Is it the number of electrodes in our EEG?
The number of electrodes, so we did two analyses, right? We did it with RNN and also EEG. So with EEG, yes, the number of electrodes is 60.
Okay, thank you.
Yeah.
Okay, thank you. And any other question that everyone has? Okay, we have 2 minutes more and no other question and great talks. And also, Frozen put the information of Twitter and link of lecturer in the chat, and more information if you wanted to link with them and ask your question in the future. And thank you all and have a good time. Bye. Thank Frozen for information and handling the panel.
Thanks, everyone.
Thank you. Bye.
Bye, everyone.
Panelists for our panel. So if you want to ask questions from each other, I don't think you can post the questions in the Q&A panel, unlike the attendees, but you can basically ask it yourself. So you can unmute yourself and ask the question from each other as well, which I encourage you to do so. So wait another 2 minutes before we will start the session. All right, so this is sharp 6 p.m. here in Lausanne, Switzerland. So hello, everybody. Welcome to this session of NeuroMatch conference. As I said, this is early evening in Lausanne, but I hope you're having a good time on any time zone you are around the world, and you stay safe. So we have an amazing lineup of speakers in this session, and we are going to learn a lot about macaques, from their behavior to neural circuits. And I just remind everyone that the session is in the format of traditional talks, meaning that our speakers would give an introduction and presentation of their work in about 12 minutes, and then we're going to have 3 minutes for questions. Feel free to ask any questions you have, and please post them in the Q&A panel, and upvote your favorite questions so they would be asked first. You can also use the chat to share any relevant thoughts or ideas or papers. And again, if there are questions that because of time we might not have time to address, then we will ask our panelists to later address those questions in the chat. All right, so with that, I would like to introduce to you our first speaker of this session, which is Ben Volo. Ben is a postdoc in the lab of Benjamin Hayden and Jan Zimmerman at University of Minnesota. And he will talk about the behavioral fingerprints in macaques. Stage is yours, Ben.
Cool, thanks for the introduction. Let me just share my screen. Cool. So thanks everyone who's tuning in for tuning in. You might be expecting to hear about the functional hierarchy of choice, but due to a scheduling change, you're going to be stuck with me. I definitely encourage you, though, to check out the work by David from our lab. It's posted up on bioRxiv, and it's super exciting. But today, I'm going to be talking about our work decomposing natural behaviors in freely moving macaques. Our ultimate goal, though, like most other people's in our field, is to understand how the brain generates behavior. And the way we typically study that is mapping brain activity to behavioral output We put a monkey in a chair and let him perform some task. And this has led to a lot of powerful insights for sure, but the behavioral repertoire that can be expressed is going to be severely limited. And this is a shame because we are throwing out a lot of key information, namely what their body movements can tell us. And consider how much information we can infer from the body language in this picture. We can tell that the guy is trying to engage the lady in conversation. He's clearly interested in her, but she is not. But if she changes her pose and now she's looking at him, well, now all of a sudden we can infer that, okay, she's interested, or at the very least, she's willing to engage with him. There's an important principle here. The expression of behavior can change independent of what the physical space is. And it's going to depend on what is relevant in that moment, what is being rewarded, what is the time scale over which we're being rewarded. And more than that, we can infer another person's internal state or motivation or intentions from the expression of their poses. And this gives us an important way to then study how the brain can organize and instantiate intentional behaviors. Now, we study this problem in macaques, as they offer a really rich source of neural data and while still having a really high translational capacity for humans. And our ultimate goal is to understand how the brain generates self-generated behavior. But to do that, we need a large corpus of behavior. Now, for various reasons, one of the best ways to do this is by immersing our subjects in large environments where they are free to move around. And this gives them the freedom to enter many different postural states over the course of an experiment. This is the setup that we're using to tackle these sorts of questions. It's a large enclosure where the monkey is free to move around naturally in all 3 dimensions, and we record them with 62 synchronized cameras. And that's important because then that allows us to accurately reconstruct their pose as they traverse through their space. And you can see the results of that reconstruction in this video. So on the left is a video of the monkey from 4 different camera views, and the colored lines are the reconstructed skeleton of the monkey, where we're tracking key joints like the neck, hands, head, or tail. And as you can see, we can track the monkey very well, even when some of the camera views have occluded joints or don't even capture the monkey. So with this setup, what we're going to do is then ask our monkeys to perform different tasks. So one of the tasks is a really classical foraging experiment task with depleting rewards. So the monkey has 4 different feeder boxes. Each feeder box has 5 rewards. Once one feeder's been depleted, there's a really large timeout. So the monkey, if it wants any more reward, it has to go to some of the other feeders. We also had one where we had a variable schedule. So some feeders had the same sort of structure with depleting rewards. Other ones offered a really large payout, but at a random time. And finally, as a control, we had a condition where there was no task at all. So same physical space, the feeders are all up, but they're not doing anything though. So the monkey's free to move around and do whatever it wants to do. So in total, we collect 18 data sets. So that's 2 subjects, 3 tasks, 3 examples from each. And we're going to split each data set into 10-minute segments to sort of trialify the data. So to get a holistic picture of the kind of stereotype behaviors that the monkeys perform in this cage, we used a dimensionality reduction technique called UMAP, Universal Manifold Approximation and Projection. So we take our high dimensional matrix of joints, reduce it via PCA to improve the computational time for UMAP, and then let UMAP learn a lower 2-dimensional embedding. And then you can think of each UMAP axis as something like a PCA axis. It tries to capture the global structure, the structure of the data rather. But the really good thing about UMAP is that it can find these embedding dimensions that can balance both local and global structures. And this is unlike, for example, t-SNE, which in a way exaggerates the differences between any local clusters to make them appear really far apart. So from this UMAP embedding, we can then generate a probability density map of each pose. Peaks in this map are stereotype behaviors that are going to occur often. And we leverage this and to use a watershed segmentation algorithm on this density map to cluster the data into iconic poses. So here, for example, we found 2 iconic poses. With all that said, this is what our embedding space actually looks like. Each of the peaks here represents an iconic postural state that is going to be visited often. And I've shown here a few examples of this. So for example, here on the left, we see the monkey's leftwards and rightward stride as it's walking. And it spends a lot of time walking, right? But there's other states where we look at it and we know exactly what they mean. They're semantically meaningful. For example, when the monkey is sitting, or when the monkey's standing and it's kind of looking out outside the cage out into the world, when the monkey's climbing along the wall, or even when it's hanging upside down. So overall, on average, our states last about 3/4 of a second. And so really this is a sub-second decomposition of behavior that we've performed. We can get even more information from this embedding space. And we can see this by looking at the transition probability matrix. So this is the probability of going from some particular state and ending up on some other state on the next time point. And I've reordered these according to the distance of these states on this map, right? And generally speaking, what we can see is that most transition happen to nearby states. We see this sort of blocky structure in the data. So that means, for example, if the monkey's spending time walking, it's going to move to another walking state often. But there's even more that we can take out from the transition probability matrix. So one expectation that we naturally have is that monkey's behavior is going to be goal-directed, or at least intentional. We can reframe that question and ask, are there hidden states that allow us to predict monkey's behavior far into the future? And we can do this by asking if the transition matrix is actually Markovian. Now, this analysis is lifted pretty much from Gordon Berman's 2016 paper. They've done really phenomenal work decomposing the behavior of fruit flies. Now, a Markovian system is going to be fully described by the transition probabilities from the current state and then the subsequent state. So if a system is Markovian, if we multiply this matrix by itself X number of times, representing X number of transitions, we're going to get something like this, which represents the steady state probabilities of each state. And you can see there's pretty much no information left, or no structure left, sorry, in this matrix. Now, another feature of a Markovian system is that our ability to predict far into the future decays exponentially. And this is evident when we can extract the eigenvalues of the transition matrix after X number of transitions, and we get a plot that looks something like this. So here I've plotted the absolute value of the maximal eigenvalue as a function of the number of transitions, and you see an exponential decay, in the eigenvalues. But the real transition probabilities actually look something like this. There's still a lot of structure in here. And when we plot the decay rate of the eigenvalue, we can see it actually decays much more slowly. And so what this implies is that there are hidden states that have a memory and that would allow us to predict the monkey's behavior far into the future. But to put it all together, what this also means is that the monkey's behavior is Markovian at short timescales and non-Markovian at longer ones. But in some ways, this is not surprising. It makes sense. If I try to predict, for example, where your arm's going to go in the next time step, I can do it really well for the next time step. But if I want to predict far into the future, I now need to have information about your goals or your reach targets and things like that, the hidden states that we can't observe. But we see the same sort of decay behavior for many of our top eigenvalues, 6 of which I've plotted here. And you can, when you compare every colored line, the dashed versus the solid versus the dashed line, we can see that the real data has a much slower decay. And so what this actually tells us is that there are multiple time scales of behavior for our macaques. But this excess decay, the difference between the Markov expectation and the observed rate of decay, does not differ by task. And so this is obviously kind of surprising, and it tells us that the task does not influence behavior in terms of the timescale of predictability. But this does not mean that behavior is actually devoid of context information. So even though every state is likely to be expressed, the relative frequency of behavioral expression is going to be subtly different. And so we can get at that by looking at the behavioral expression profile. So this is the frequency with which a monkey is going to visit any particular state. And this kind of approach is has been pioneered by Bob Dada's lab, who've also done a lot of really great work in decomposition of mouse behavior. And recently they actually published a paper in Nature Neuroscience looking at this expression profile as a function of drug conditions. So what we're going to try to do is decode the task as a function of this behavioral expression profile. And indeed, we can actually predict which task generated this data with 76% accuracy, above what we'd expect from a random shuffle of our data set. And we can also predict the subject with 99% accuracy. So our subjects are extremely idiosyncratic in how they move and express their behavior. And with that, I'm ready to conclude. So we found that we've leveraged a method, postural embedding, that can actually preserve meaningful semantic structure in our data. And we found that the behavior is Markovian at short time scales and non-Markovian at longer time scales. So sort of giving us now a handle on, starting to give us a handle on the hidden states that we can leverage to ask questions about our neural data. The predictability of behavior does not depend on the task, but the actual expression of behavior does. And so these kinds of results are really a really crucial stepping stone to the kinds of questions that we want to ask moving forward, where we want to look at more fine-grained analysis of poses that can predict the task, relate behavior to the actual different task elements themselves, and finally actually map all this to the neurons. And so with that, I'd like to thank everyone who's been really critical in this project. It's a really large project involving a lot of people in the lab. My principal advisors, Jan Zimmerman and Ben Hayden, been a fantastic source of support. Hyunsu Park, who's a collaborator with us, has been really critical in actually getting the pose deconstruction done, as well as Praneeth Bala, David Basin, Jen Holmberg, and Runal Zambre, who were also involved in the day-to-day operations of the cage. And with that, I'd like to thank you all for listening.
Great, thank you very much, Ben. We went a little bit above time, but it's all right. On behalf of all the attendees, I'm gonna have you give you a set of round of applause. Yeah, very nice talk. And it seems, yeah, I mean, I think there's a collective attention more and more in the field towards understanding better and better and quantifying in more details behaviors. As you mentioned yourself in rodents and now monkeys and I guess also humans, if you want to understand better the neural circuits. Yeah. So I don't want to take so much time, so I'm going to ask the first question that we have from the audience. Malcolm is asking, is the eigenvalue decay rate at all dependent on how many UMAP dimensions you use? Have you tried more than 2, for example?
Yeah, that's actually a great question. I've got a figure for this. So one thing that we can do is a sort of naive reclustering. Instead of, we take our 31 states, our embedding space, and then try to recluster it into different numbers. This is kind of what the clusters look like. And then we look at the excess decay, right? And what we see is actually it pretty much doesn't matter. Only if we get down to 3 clusters or lower will we get a difference in decay rate, or a smaller difference in decay rate. But still, it's above chance. So it's still non-Markovian, but it does look to be a little bit faster with less clusters.
All right, great, thank you. I had one more question. I mean, again, maybe a basic one. I think you mentioned that briefly, but I did not really capture that. So what is the difference between these UMAPs and the TSNE maps, and what are the advantages of one representation over the other?
Yeah, well, so what TSNE does is it exaggerates differences. So if there's going to be natural clusters in the data, what it's going to do is actually try to pull them apart as much as possible, so that you can visualize and analyze them better. But that means that we don't have a way to actually map distances, like distal clusters, right? The distance between them is not a meaningful number. And so UMAP actually has a parameter that lets you balance the sort of preserving the local structure versus the global structure. So it gives you a more kind of, I guess, holistic view of what the actual data space looks like.
All right, great answer. Thank you very much again, Ben. And for the interest of time, we have to move to our next talk. Thank you very much again. All right, so our next speaker is Alessandro Bongiovanni. He's a postdoc in the lab of Matthew Rushworth at University of Oxford. And Alessandro will talk about the role of medial frontal cortex in a novel inferential choices. Stage is yours, Alessandro.
Thanks. So I will share the screen. Do you see? Okay. So yes, as you said, I'm going to present this work that is just about to be published, and it comes from my PhD thesis. So the key question we want to ask is whether there is a neural circuit that is specific for making novel inferential choices as compared to familiar choices, for example. And we also want to bridge the knowledge that we have that comes from the neuroscience of value-based decision-making in the monkey and in the human, because you probably are aware that people studying value-based decisions in monkeys have generally focused on the orbitofrontal cortex. So coming from Padoa's Schioppa and more recently other studies have found that neurons in OFC correlate with the values of the options that are presented and allowed to make the comparisons. Lesion studies by Murray, for example, and other labs have found that disruption of this area will impair decisions. But when you take a human and you do an fMRI study, you generally find that economic choice activates a difference area, which is often called VMPFC, or in any case, medial frontal cortex extended to ACC. So is this a difference of species, or is a difference of task or a technique used and so on? This is a bit the question that we wanted to ask. These two areas are sometimes confused as if it was a single one, but if you look at anatomical studies, they have different connectivity profiles, with the medial frontal cortex being connected with the rest of the medial part of the brain, including cingulate and medial temporal lobe, whereas the OFC has a different profile. Now, you could guess that a monkey study is not quite the same as a human one, because the animal received a long training, and therefore its learning is gradual. So it will sort of slowly acquire the different elements of the task and the associations. So maybe this is what OFC is about. Whereas a human is generally taught the instructions for the task just minutes before entering into the scanner. And so the learning is much faster, and the task is performed for the first time at the time of the experiment. So maybe this is what the VMPFC is about. In order to test this hypothesis, well, let me explain better what I mean by novel choices with this example. So if you need to go on holidays and you can choose between these two beautiful Italian cities, but you have never been to either one, you can choose between Siena, which is a medieval town, is in the sort of on a hill area, whereas Syracuse is more in the south, it's on the seaside. So you can sort of make up your mind about the value of each option, even if you don't know them, by putting together the different attributes. So you may know whether you like or not the seaside, you may know about the food and so on. And so you create a compound value, and then you make your choice. Of course, this is not the case in 2020 because of the coronavirus, so you shouldn't go on holidays probably in the next few weeks. But to summarize, yes, we ask whether monkeys can perform these novel choices, whether they are recruiting a different neural circuits, and later we'll also ask whether this circuit is encoded by a grid-like coding that has been extrapolated from what we know about navigation in physical space. So the task works like this. You have a first training set where the monkey for a long time is trained to make binary choices between options that associate different colors with different amounts of juice. Then on separate days, they learn to make binary choices again. And now the options are associated with different probability of rewards. But know that in every case, one attribute is changed and the other is fixed. And so these two training sets form this sort of gray area in the more general space of all possible combinations. And so you can understand that the monkeys will potentially know about all the other areas in the sense that they know the meaning of the color and the meaning of the dots, but they have never been trained on these white spaces, so they've never been reinforced by choosing this specific combination of color and dot. We have three different experiments. The first one when the monkeys make a binary choice into an fMRI scanner, so we can see the neural activity. Then the monkeys will do a single sort of response task where there is no choice. And this we use for addressing the question of what are the neural representations that underlie these novel options. And finally, outside the scanner, we disrupt the behavior using ultrasound, and we see whether the effect on the brain makes a difference in the responses. So to answer the first question, the monkeys can actually make these binary choices. So here on the right, you can see a decision pattern of 4 different monkeys. And you can see that when the value of the two options is close to each other, they are indifferent. But when one option is much better than the other, they choose the best option. So this is the case for novel choices like here that are visually similar to a familiar choice. But this specific combination has never been performed in the training phase. Now, what happens at the neural level? First, we put all the choices together, familiar and novel, and we use this regressor that is the chosen value minus the unchosen value. So we see whether the neural activity is modulated trial by trial by the difference that of the two options, which is the amount of value that we believe should drive the comparison, of course, because the task is to compare the two. And you can see that, quite unsurprisingly, orbitofrontal cortex, which is the area where we know that neurons code value, has an activity for the difference between the two options. However, now we do another contrast, which is value difference in novel trials minus value difference in familiar trials. So we see whether the two different types of trials activate differently the brain. And we see that they do so in medial frontal cortex. So in OFC, the effect is similar for familiar and novel, but in MFC, the effect is maximally different between novel and familiar choices. So we argue that this MFC circuit has been recruited specifically by these novel choices, where the monkeys know the components of the options available, but has not a very long training that has sort of strengthened the association by reinforcement. Okay, so then we are asked, what are the neural representations that underlie these novel options? First, we use a repetition suppression design. I go quickly on that, but the design means that when you show the same thing twice, the brain will respond less on the second time, so there is a suppression. And we see that when you present the same stimulus twice, you have an activation in orbitofrontal cortex again, which is sort of similar to what we got before, a bit more anterior. And so we assume that this area encodes the identity of the options that are presented, but not the value independently of their identity. Now, in order to ask how this value is computed, we took this hypothesis for the literature on grid codes. So if you remember, when a rat, for example, navigates into a space, you will have some place cells that will activate, will have a receptive field in just one location. But then they will have a grid cell, usually in entorhinal cortex, that have an hexagonal pattern of symmetry in the receptive field, which means that the same cell will activate in many locations, but not all over the place, with a very specific structure. And now we want to see if this spatial value space that we described can be analyzed in the same way. And we found that there is indeed a signature in the fMRI that, well, the data are a bit noisy because we have very few choices before the choice will not be novel anymore and will become familiar again. But when you look at novel choices, you have a peak of activation precisely in this medial anterior region of PFC. And we test that this effect is specific for a six-fold symmetry. So the pattern is hexagonal and not for other types of periodicities. And we also tested by estimating this grid in two separate subsets of each session, there is a consistency. So we now know that in MFC, there seems to be a grid code that spans this non-physical space that monkeys are exploring when they see one option at a time. So they essentially navigate into this space, even if it's an abstract space composed by the amount of reward and the probability of rewards. And then to conclude, we decided to disrupt this network by using transcranial ultrasound simulation that can be done without invasive surgery, but is equivalent in some sense to TMS, but can reach more deep brain areas. And by doing this, we asked the question of how the monkeys is going to combine the two attributes. So you remember that you have magnitude and probability, and the correct way to do is to multiply magnitude and probability and to estimate the value of, to choose the option that has the highest magnitude time probability value. However, you can take an easier way, heuristic, and you just sum the magnitude and the probability. So this is what you do whether you are unable to integrate the two properly. And we compared how much they follow one strategy and the other, and we noticed that when you stimulate this more anterior and central part of the PFC, you have a specific decrease in the amount of optimal choices made by the monkeys. In this case, we have only 3 subjects after stimulation. So to sum up what we have seen, MFC disruption impairs ability to base choices on integrated value, and therefore they rely more on the 2 separate dimensions.
Alessandro, yeah, okay, great. If you can wrap up in a few sentences would be great, thanks.
So you have the first experiment when we show that they can do these novel choices, and MFC has a different effect for familiar novel choices, even when you keep the difficulty the same. The second experiment showed that when you show one option at a time and you move around the space, you activate a sort of grid code. And the third experiment shows that when you disrupt this network, then the ability to make optimal choices is indeed impaired. I need to thank all my lab, and in particular my supervisors, Matthew Rushworth and Mira Klam-Fluger, as well as the other co-authors of the paper, David Leonard and Jerome. Thanks.
Thank you very much, Alessandro. Again, on behalf of all the audience, great talk and very interesting data again. So while again, I wait for the questions to come in the Q&A panel. So I found this like homology between the coding scheme you find in MFC and this grid cell type of presentations, quite fascinating.
Yeah.
But to admit, I have to say it wasn't very clear. Maybe you had to a little bit go fast on that. It wasn't clear to me how did you interpret it as actually as grid presentation somehow. Could you go back to that slide if possible, maybe just to.
You asking how you do the analysis to test?
Yeah, I mean, how do you interpret that as a a grid type of coding?
So maybe I can. Do we have a? Yeah. well, I don't have an image for that, sorry. But anyway, so here the idea is that when you move into this two-dimensional space, you occupy different locations, right? And the options are succeeding each other very fast. Now, there is supposed to be a symmetry in the effect. So the only problem is that you don't know what is the orientation of the grid. So we do what is called an F-test, where you have the sine and the cosine of the trajectory. So at every single trial, we estimate this angle alpha, which is how you go from the previous option to the next option. And if either the sine or the cosine of this angle multiplied by 6 modulates the neural activity, it means that the neural activity goes up and down depending on whether your direction is aligned or misaligned to the grid. So if you imagine going this direction, then this, can you see the mouse? Then you go.
Yes, yeah.
Every time you move along the grid, you will have maximum activation. And every time you move sort of obliquous, then you cross the white space and will have lower activation. So when you try all possibilities, you will see this 6-fold symmetry that goes up and down 6 times within 360 degrees. That's the way we tested based on previous papers. I think the first one of which was by Christian Doller.
Great, thank you very much for the great answer. And I was just wondering if you plan, given that, I mean, I think, can you compare that, I mean, you started by comparing this behavior of novel choices to human. So one of the advantages that you have in macaques is that you can also go a little bit more in details. And are you thinking of other techniques, like really looking at then seeing your neural recordings and see if the FC can also see this type of coding at the level of single neurons or population of neurons?
I think, I mean, I didn't perform electrophysiology in the past, but this is one of the avenues we're exploring. I think that would be particularly interesting to have single unit recordings in MFC. And there are few people who publish results there. Many people say that the activity is relatively small compared to what you can measure in OFC, but probably they didn't try our task, so it would be a good thing to do.
Great. Thank you very much, Alessandro, again. And again, for the interest of time, we have to move on. And thank you. Our next speaker and our last speaker of this session is Ben Dean. Ben is a postdoc in the lab of Weinrich Freywold at Rockefeller University. And as you see, he will tell us about brain regions in macaques, again, related to social reasoning and memory. The stage is yours, Ben.
All right, thanks. Can you guys hear me? Great. So I first want to acknowledge my co-authors on this project, Sophia Landi, who's a co-first author, my advisor, Weinrich Freywold, others in the Freywold's lab that helped out with animal training and data collection for the project, and our various funding sources. So as humans, we have a rather sophisticated ability to understand and predict the behavior of other people, and we do so based on their internal mental states, as well as our knowledge about aspects of their personality, so personality traits and personal history. And these abilities are thought to be supported by a distributed set of brain regions across association cortex, including the temporoparietal junction, precuneus and posterior cingulate cortex, and several regions in the medial prefrontal cortex. These regions fall within a network known as the default mode network or apex network. And in addition to responding to a range of high-level social cognition tasks, these regions appear to be involved in storing information about familiar people. So if you ask humans in a scanner to think about specific familiar people, spatial activity patterns in these regions can be used to decode which person they're thinking about. And importantly, these regions don't comprise the entire default mode network, but rather constitute a subset thereof. So in humans, based on functional connectivity and task data, you can find multiple interdigitated subnetworks of the DMN, one of which is involved in social cognition, the other which has been argued to be specialized for episodic recall, and by others to be specialized for spatial reasoning or navigation. So in this talk, I'd like to discuss the evolution of this specialized system for social cognition in humans. So in terms of the default mode network, studies of functional and anatomical connectivity have suggested that default mode networks exist across a range of mammals. They've been found in macaques. They've also been found in rodents, both rats and mice, which diverged from us over 80 million years ago. That said, it's much less clear whether these regions in non-humans play any role in social cognitive function, or whether there's anything like socially specialized subregions or subnetwork within the default mode network in non-humans. So I'm going to ask this question by studying macaques. We do so because macaques are a social species. They recognize others based on their faces and voices, and the extent to which they have human-like high-level social cognition is debated and has been debated for the past 4 decades or so. But at least to my mind, there's compelling evidence that they at least understand basic mental states of other primates, things like percepts and desires, and make action predictions based on those. So you can imagine various hypotheses about social specialization within the DMN in macaques or in a common ancestor. So one possibility is that there's no specialization, so that these animals have a default mode network, but they don't have socially specialized regions within that. Sort of the other extreme, we can imagine that macaques might have a fully human-like social subnetwork of the default mode network. And we can imagine a range of possibilities in between, in which maybe one or several subregions of the DMN have begun to evolve socially specialized components, but they haven't propagated throughout the full network. So we're going to test this using macaque fMRI. This is a powerful method, as Alessandro mentioned, for sort of searching for specific functional signatures across the entire macaque brain. However, macaque fMRI isn't particularly suited for studying sort of naturalistic social interaction or social behavior in monkeys. You have monkeys in a small tube, head fixed, looking at a screen. That being said, what we can do relatively easily with macaque fMRI is have them fixate and show them images, images of faces and non-faces. So here we're going to capitalize on the fact that in humans, these regions don't just seem to respond to high-level social cognition tasks, but also, as it turns out, respond when you just perceive an image of a familiar face relative to an unfamiliar face or a familiar or a non-face object. So specifically, we showed macaques 6 categories of images: personally familiar faces and objects. So these are faces and objects that the macaque has personally experienced with and interacted with. Visually familiar faces and objects. So these are faces and objects that the macaque has no personal familiarity with, but they've seen the specific images before. And unfamiliar images, which the macaque has never seen before. This was data collected by my co-author, Sophia Landi, as part of her graduate thesis. All right, so when we compare responses to faces and objects in macaques, this is showing an example from one monkey. We see sort of the expected set of regions along the temporal lobe following a progression or a hierarchical progression from posterior to anterior parts of the temporal cortex. We also see the sort of well-known regions of lateral frontal cortex that respond to faces in the macaque, PO, PV, and PA. When we look on the medial surface, we also see a set of regions within medial prefrontal cortex that respond to faces over objects. And we sort of see what looks like an alternation of face-preferring and non-preferring domains of frontal cortex along a dorsal to ventral axis. So this is in one monkey. If I zoom in on the MPFC and look at results in the other animals, we see a consistent result in the second animal, the third animal, and the fourth animal that we scanned. Okay, so we've identified a set of regions in medial prefrontal cortex that appear to have some social specialization in that they respond to faces over objects. We then wanted to compare the anatomical and functional properties of these areas to what we know about corresponding social cognition areas in humans. So we'll do so by comparing their anatomical organization, their response profile across multiple categories, and their functional connectivity in the resting state. So in terms of anatomical organization, there's sort of immediately a striking parallel with the organization that's been observed for human social cognition regions, which is that in both species, you see this sort of alternation of socially preferring and non-preferring domains across the dorsal to ventral axis in MPFC. We next wanted to ask, is this a pattern that we're sort of reading into the data into a noisy response, or is that actually a reliable pattern of response in our data? So to do so, we split our data in half. In each animal and hemisphere, we can define at least 3 subregions of MPFC that respond to faces over objects. So we define the peak coordinates of those regions, and the peak coordinates are the coordinates of two non-face preferring areas between those three regions. Then draw splines between those coordinates, and we extract responses to faces versus objects in left out data. And this is what we see. In the left hemisphere in our left out data, we see a consistent alternation between face preferring and non-preferring zones of MPFC, and a similar pattern in the right hemisphere. So this pattern of alternation appears to be a reliable pattern in our data, both across halves of the data, across hemispheres, and across animals. All right, we next want to ask about the response profile across multiple conditions in the experiment. And our expectations from human research are one, that we'll see a response to familiar faces over unfamiliar faces, and two, that we'll see a deactivation to non-preferred stimulus categories, namely objects. And this is something that's been observed in prior human studies focusing on socially responsive regions of MPFC. This is an example from a social cognition task, but also is characteristic of other regions in the default mode network that they tend to deactivate relative to baseline to non-preferred stimulus categories. All right, so to do so, we do a leave one run out ROI analysis. So we take n-1 of our runs, extract the top 5% of face responsive voxels within a search space, and then extract the responses of that region across categories in the left out run to avoid a non-independence error. And I'll first show you what this looks like for regions of temporal cortex. So this is a search space consisting of area TEO, which contains face area ML. When we apply this analysis to this region, this is what you see. So you see a strong response to all the face conditions over objects, about a 4% signal change dynamic range, and a slight preference for familiar faces. Okay, we'll now look in MPFC. So we defined 3 search spaces for MPFC, spanning ventral to dorsal MPFC, defined based on Brodmann areas. When we extract responses in ventral PFC, this is the pattern we see. So the strongest response is to familiar faces, sort of a low to baseline response for unfamiliar faces, and a deactivation to objects. And we can test the face and familiarity effects using a linear mixed model with animal included as a random effect. We find that these are both highly significant. In middle medial prefrontal cortex, we see a very similar pattern of results. And in dorsomedial prefrontal cortex, we see a similar pattern, although somewhat weaker, and an overall deactivation relative to baseline. So this suggests that these socially preferring regions in macaque and PFC have somewhat similar functional profile to corresponding regions in humans, in that they prefer familiar to unfamiliar faces and deactivate to objects relative to baseline. All right, lastly, we're going to look at the resting state functional connectivity of these regions in macaques. So based on human data, we expect that these regions should be functionally connected to other parts of the default mode network. And in human data, you also see different patterns of functional connectivity for socially preferring and non-preferring regions within MPFC. So you see a different pattern of connectivity within the default mode network. All right, so we'll first look at the pattern of functional connectivity for a ventral face preferring seed in macaque MPFC. And this is the pattern we get. So you see a clearly default mode network-like pattern of functional connectivity, including other regions in the medial prefrontal cortex, posterior cingulate cortex, parahippocampal gyrus, dorsal STS, and an intriguingly TPJ-like region of lateral parietal cortex in Brodmann area 7A. And when we then look at functional connectivity of an adjacent non-socially preferring region of MPFC, we again see a pattern that looks much like the default mode network, but this pattern doesn't differ significantly or substantively from the pattern that we saw from the socially preferring seed. So we find that these regions of MPFC do fall within the default mode network in terms of functional connectivity, but we don't find any evidence of distinct subnetworks within the DMN in other parts of the default network in macaques, unlike in humans. All right, so to summarize, we've identified a set of socially preferring subregions in macaque and PFC. We find that these regions are similar to corresponding human areas in terms of anatomical organization, response profile across multiple categories, and resting state functional connectivity. And getting back to our hypotheses, these results, we think, rule out the hypothesis of no specialization. They indicate that there is some early social specialization in the macaque. They're most consistent with a hypothesis of partial specialization, in that we only find this within MPFC, not in other parts of the default mode network, and we don't see evidence for distinct subnetworks of the default mode network throughout association cortex. That being said, we can't rule out the possibility of a fully human-like default or social subnetwork of the DMN. It's possible that we might find this with something like a more active social interaction or social cognition task, or with more sensitive methods like electrophysiology. All right, thanks for listening, and I'm happy to take questions.
All right, thank you very much, Ben. Again, sort of applause for you, and again, on behalf of everyone, all the attendees. Very beautiful talk and very clear. Again, while I'm waiting for the questions, so I had the first, let's say, I mean, again, very beautiful technique that you can, as you said, like really visualize the activity of whole brain using this fMRI technique. One question that comes to mind is, can you think of, let's say, a behavior task where you could, and also other techniques where you could also test the causality of these networks, like for social behavior? Have you thought about, or is that in your plan, let's say?
Yeah, that's a great question. I tend to think that these regions would be involved in social decision-making tasks. So various people, various groups have implemented sort of economic game tasks with non-human primates. There's a question as to what extent those tasks are really sort of eliciting social cognition or sort of social inferences in monkeys. But insofar as they are, or insofar as you can design them such that they are, maybe have another monkey present in the room with the monkey you're recording, I would expect these regions to be involved in that sort of task. But yeah, it's a great question and something that we're actively thinking about.
Yeah, I was a bit thinking in those lines. For example, now I guess the group of Michael Brecht, they are doing like tasks for rats, I think, and then they're also studying, let's say, social behaviors and like approaching two rats, or I think it's in rats if I'm not mistaken. They're approaching two different rats to each other while they're tracking all their interactions to each other, and then while they're recording neural activity from regions that, again, they think they might be involved in social behaviors, and they have beautiful data in that. And again, I guess thanks to the technologies that we also heard and about, actually in this conference, about all these pose estimations, automated pose estimations, and so on. Again, if one could imagine, as you said, rather than just images, bring in other monkeys and then try to somehow look at those interactions would be, I think, amazing. I do not see any questions in the Q&A panel. So I think with that, I would thank you again for the great talk, and again, all our panelists, Ben, Alessandro, and again, Ben. So we had two men in the same session. And because we are running now out of time, and I would like to thank also State Art, which was our back end for running smoothly this session. And thank you very much, all the audience, for your participation. And I hope everybody enjoys the rest of the conference. Thank you very much.
Thanks so much.